Awesome. Well, thank you everybody for joining us today. As you can see, we have Dr. Carrie Clark. Dr. Shirley Hartman is running a little bit late. She got caught in traffic, so she should be joining us shortly via this webinar, but thanks again for everybody for joining us today. I also want to take a quick second to shout out actually first Melissa Bell, who you have just seen, and Florida Lyme Disease Association, as well as Bay Area Lyme for their partnership. None of this would be possible without them. And I know a lot of the research that Carrie Clark is doing, as well as some future stuff, has been made possible from both Florida Lyme, Bay Area, and then also ourselves. We were very happy to have recently given some funds to Dr. Clark as well. So let me quickly introduce him. Dr. Carrie Clark is an epidemiologist and professor at the University of North Florida. He has studied Lyme disease and other tick-borne diseases in the southeastern United States for the past 20 years. His studies have involved ecological investigations of Lyme, Borrelia, invertebrates, and ticks, as well as studies of tick-borne infections in humans. Much of his work in the past decade has been focused on developing molecular detection methods for Lyme, Borrelia, and other tick-borne disease agents. He was one of the first to identify Lyme disease bacteria in naturally infected lizards in both Florida and South Carolina. And he was also the first to identify Borrelia americana and Borrelia andersoni infections in human patients in the US. His work is demonstrating that Lyme disease is much more common in the southern US than previously thought, and that Lyme disease in the US is associated with multiple species of Lyme Borrelia. Clearly, he is a great person to be on this Lyme in the South webinar, and we thank you for joining us today. Looks like we also just had Dr. Hartman join us. Dr. Shirley Hartman is the owner-operator of Mandarin Wellness Center based in Jacksonville, Florida, which she has founded in 1987. She obtained her medical degree from the University of Iowa. Over the years, she's developed experience treating people with fibromyalgia, chronic fatigue syndrome, and chronic pain. When she learned about Dr. Clark's research, they also have a relationship here. She began sending him samples from her own patients to test. An amazing percentage of the people with several symptoms within the spectrum of pain, chronic fatigue, fibromyalgia, and neurological disorders showed evidence of tick-borne disease. So again, thank you, Dr. Hartman, for joining us today. Both of these guests are really well-versed in Lyme disease in the South and should do a great job of conveying that to all you attendees today. So I'm going to go ahead and just jump right in here and introducing ticks and disease in the South. So I'll start with you, Dr. Clark. If you could explain what types of ticks live in the South, and I know you mentioned you have some diagrams, so I'm very happy to have you share anything that might be helpful for our attendees to learn about ticks in the South.
Okay, well, in that case, thank you, Noah, and appreciate the intro and the opportunity to meet with everybody and to also meet with Dr. Hartman today. It's nice to have her as well, and look forward to having a fun time this evening talking about some tick-borne stuff. So if I can share my screen here, then I may pull up well, I've got several files here, but I do have a few that talk a little bit about some of the different ticks in the Southeast. So I'll try to answer that question about ticks in the Southeast. Can you see that screen there?
Yep.
Okay. So, you know, we have numerous species of ticks in the Southeast, but there are certain ones that are more important than others. And of course, the ones that bite humans are the ones that we're most interested in. And what you see here are just some images of some of the adult stage of several human biting ticks in the southern United States. So starting up here at the top left, you see an image of an adult male Lone Star tick, an adult female Lone Star tick. You can see why the female is called the Lone Star, because she has that single white dot on the dorsal surface. A lot of people misidentify the male because it doesn't have that single white dot. It's just got this pattern. And that's the most common, abundant, aggressive human biting tick species we have in the Southeast. And the majority of tick bites that people get, probably 90+%, will be some life stage of the Lone Star tick. And all of the motile life stages will bite people. So the larvae will bite people, the nymph stage will bite people, and the adult stages will bite people. Over here on the top right, you have the black-legged tick, which people often refer to as the deer tick, but the actual name is the black-legged tick or the eastern black-legged tick. And this is the adult male on the left, the adult female on the right. And those adult stages do bite people, depending on what part of the southeastern United States you're talking about. Their behavior is a little different. So in the more northerly areas of the South, the nymph will actually bite people. When you move further south, the nymph stage does not typically bite people because it tends not to seek a host above ground level. And I think that's primarily due to the ecologic conditions in the South, but the adult stages do definitely bite people. They're more active in the fall and winter and early spring. But you can collect the immatures as well in some parts of the South. And in fact, I just got back from the mountains of North Carolina a few days ago, and I actually was able to collect some of those on a cloth drag. I may show you a picture of that later. On the bottom left here is the American dog tick. That's the primary vector of Rocky Mountain spotted fever in the eastern US. And this is the adult male and the adult female. And then on the bottom right is what's called the Gulf Coast tick, which is another Amblyomma species. So it's in the same genus as the Lone Star tick. They're more scattered in distribution. They will bite people when you come into contact with them, but they're not biting people at the same rate that some of these other ticks are. And then in the very middle here is something called the brown dog tick, which is a species that's found all over the entire world, but it's not usually found in nature. It's usually only found around humans and their dwellings, and it's very common around kennels, dog kennels, but it will actually get inside people's homes sometimes and can actually cause an infestation in people's homes. And they will readily bite people if they get an opportunity. And they've been known to transmit Rocky Mountain spotted fever. They can probably transmit some other pathogens as well. It just hasn't been proven yet.
So Dr. Clark, that's extremely interesting. I think a good follow-up to that is to talk about the types of hosts that these ticks live on or will inhabit.
Yes, that's a very good question. So one of the things that we know about most of these tick species, and I can pull that slide back up here, is that these ticks are not extremely host specific. They tend to feed on smaller animals in their immature stages. So when they're larvae and nymphs, they often feed on smaller animals like rodents, like mice, rats, squirrels, and things, and birds. And Lone Star ticks will feed on primarily birds, and they'll even feed on deer in the immature stage. They don't feed on rodents very much, at least not in the southeastern US. But in the adult stage, they definitely feed on deer, and deer are important for the numbers of the ticks. For most tick-borne pathogens, deer are not a reservoir, so the ticks don't get infected by feeding on the deer for things like Lyme and some other pathogens. But the deer are incredibly important for the numbers of the ticks. So where you have high deer density, you tend to get higher numbers of ticks for some of these species. And that's definitely true for Lone Star ticks. It can be true also for the black-legged ticks. And with them, again, immatures will feed on birds, they'll feed on small mammals of different types. And then the adult stage will feed on larger, like medium-sized mammals. And then they'll also feed on deer. And again, as I mentioned, they'll feed on people. So the fact that they're not real picky about what they feed on is probably one of the reasons why they readily bite people. With the American dog tick, important hosts for the immatures, again, are small mammals. It can be anything from mice to rabbits, squirrels, and so on. And then in the larger, in the adults, they feed on things like raccoons and dogs and coyotes and foxes and maybe bobcats and other animals, sometimes larger animals as well. And then Gulf Coast ticks, immatures will feed on birds and really a variety of different animals. They often feed on birds, but will also feed on other things. The adults will feed on larger sized mammals, including cattle and dogs and cats and so forth. And then the brown dog tick, it feeds on mostly dogs, but when people are around, or if a person's residence gets infested with these things, they will readily bite people. And occasionally I get calls from people who ask me, about some issue they're having, and they say, my house is infested with ticks. If the house is infested with ticks, it's almost certainly the brown dog tick. It means that somewhere along the way, their dog got a female tick on it, somewhere, usually at a kennel when they boarded it, or maybe at a dog park. They bring it home, the tick feeds, drops off, lays eggs, and this tick can go through its whole life cycle inside the home, and people's homes sometimes get infested. And when that happens, then usually people in the house start getting bitten.
That's some really interesting information. I think one of the biggest takeaways there, and this whole webinar is to dispel some myths about Lyme disease in the Southeast, is that I know general consensus, if you talk to people on the street, they're going to assume it's only deer that are hosts. And I think you made a clear point that that is not true. And that's why, sometimes these cases can spread farther than expected. And I think that's a good transition into my next question, which is, do you think that the CDC and Florida Department of Health statistics on Lyme disease in Florida are accurate? And if yes or no, why or why not?
So the easy answer on that is no. And at first I would simply say, for the same reasons that they're not accurate anywhere else. The case definition is written purposely to be incredibly strict, and that means it makes it very difficult to meet the case definition to be called a confirmed case. And that is true all over the country, and that's why the estimates of the actual numbers of cases of new infections per year range up to as high as 400,000 new cases a year. That's not being reported, but that's just based on estimates from and medical data, people actually treated for Lyme. And that's one of the negatives of using a super strict definition, is that it underestimates the true incidence of the disease. And I understand all that. I'm an epidemiologist, so I know the value of a sensitive case definition versus a strict case definition and so on. But it severely underestimates the magnitude of the disease. So in Florida, I think we have a mixture of things going on, and I'm sure this is true for elsewhere, where we have a lot of people move to Florida from other parts of the country, and where Lyme is probably very prevalent, and they get infected and they move to Florida, and then they get diagnosed here, or they try to get diagnosed here. Let's say they have onset, or they recognize that they have something going on after they move here. And then sometimes they're fortunate enough to get diagnosed. Maybe they're fortunate enough to meet Dr. Hartman or someone like her who's very knowledgeable, and perhaps get some help that they need. But also we have endemic Lyme. So the state of Florida considers Lyme to be endemic in the state. So I appreciate that they acknowledge that, but the application of this super strict case definition will minimize the number of cases that get recognized as Lyme and that go into the official disease statistics. And then it artificially makes the disease look to be very rare. And I think it's a lot more prevalent than the numbers indicate. And the same is true for probably almost every tick-borne infection that occurs here.
I have to agree, because the CDC case definition is far too strict. And I just see patients every week who didn't meet the CDC criteria for the diagnosis of Lyme, but they have Lyme. They have symptoms consistent with Lyme. They do have some evidence, most of them, of antibodies to Lyme, but they don't meet the CDC criteria. They hurt when you give them treatment for Lyme. They have symptoms consistent with it. They improve when you treat them for Lyme. I honestly think it's hugely underdiagnosed in the South, and it creates a massive problem because people will go to see an infectious disease doctor and they'll be told, well, there's no Lyme in the South. You can't have Lyme. I don't know how many people who actually met CDC criteria that I've seen who were told by the infectious disease doctor that they couldn't have Lyme because there isn't any Lyme in the South. A couple of years ago, Quest Labs published data on the incidence of CDC positive Lyme that was found in various states, and Florida was one of the fastest growing states in terms of the number of CDC positive cases of Lyme. If you look at statistics of Lyme reported on dogs, there were something like 6,000 a year in Florida. How you can have that many cases of Lyme in animals, but there's no Lyme in the ticks that would bite people. It just doesn't stand the logic test. And, the bottom line is when it's so hugely underdiagnosed, we have all kinds of people that go years and decades with severe Lyme that just escalates without any treatment. Lyme also has the ability to suppress the immune system. One of my favorite articles that I hand out says the immune system cannot generate immunologic memory when there's infection with the Lyme disease agent Borrelia burgdorferi. So it suppresses the ability of the immune system to recognize Lyme and form antibodies to it. But that's how we test for it, is with antibodies. So, it becomes a vicious circle. We don't get test positive cases. The CDC and the Infectious Disease Society and the infectious disease specialists say there's no Lyme here. And then people just keep going on being sick, untreated. There was a study that was published a number of years ago where they analyzed the quality of life in people with Lyme, and their quality of life was worse than people with severe congestive heart failure and multiple sclerosis. That's how sick many of these people are. And they're just going around untreated because everybody tells them they couldn't have Lyme.
And something else that's really important about this, Noah, and all of our viewers, is, even if Lyme disease, the bacteria that causes Lyme, was not or maybe we would say enzootic, even if it didn't in nature in Florida, we would still have lots of people in Florida who have Lyme because of travel from other places. And yet, there seems to be no acknowledgement of even that possibility. And once someone moves from one of those hyper-endemic areas, it's as if the Lyme can't cross the state lines with the human being, and those people can't even get diagnosed most of the time. And that's, so imagine what it's like when you have no travel history and things like that.
Yeah, I think you both just did a great job of setting up the next section of our event here, which is describing issues facing patients. Dr. Clark, I want to relate back to a presentation that you gave at International Lyme Associated Diseases Conference that outlines some of the false generalized statements that are misleading to residents, doctors, and visitors to the state and cause issues for patients. Those are, first, that Lyme is rare to non-existent in the South, which you both clearly just stated is incorrect. Second is that Lyme just doesn't exist at all, that there is starry southern tick-associated rash illness, but the diagnosis has no available tests, treatments, or research, essentially. And in addition, only deer ticks, which is something that you've also outlined already, can transmit Lyme, and that those ticks don't exist in the Southeast. Only Lone Star's ticks do, and they, for whatever reason, apparently cannot transmit Lyme disease. And then the last one is that positive test results are false positives. These are all things that patients hear on a regular. So to bring this back to the both of you, I would ask to describe kind of the biggest challenge that you hear for patients. I know Dr. Hartman, you treat them directly, and Dr. Clark, you've spoken and interviewed with many through your research. I'll start with you, Dr. Clark, if you could outline what you would see as the biggest challenge facing patients.
Yeah, thank you, Noah. So, yeah, those are a lot of things, and that's only some of the list, right? So, one thing I've learned about Lyme and other tick-borne infections is that just a little bit of knowledge can be really dangerous to patients, especially when that little bit of knowledge is related to the clinician. I've heard other similar, myths, like, for example, only the white-footed mouse is a reservoir for Lyme that infects ticks, and if you don't have the white-footed mouse, you can't have Lyme. That's another one that I've heard patients relate from their physicians. So they may have read some paper that was 20 years old or something about that. So, the first thing I would say is the tick that transmits Lyme is the eastern black-legged tick, often referred to as the deer tick. It is the same species over the entire eastern United States. Now, there are some genetic differences between the northern populations and the southern populations. That's been shown. And there are behavioral differences in the ticks, as I mentioned previously, where the further south you go, the less often you run into the immature ticks on vegetation. So they tend not to bite people as often as you go further south. This is an ecological thing because these immature ticks are very fragile. They need high humidity, and they don't survive well in really hot, dry areas. Now, yeah, it's real humid in the south, but it's really hot as well. And we have very sandy soil, and the soil dries out quickly when it's exposed to the sun, and these ticks don't survive as well. So they don't quest, if you will, seek host above the leaf litter in the immature stage. But the adults of the black-legged tick do transmit Lyme. They've been demonstrated in the laboratory to be perfectly capable of doing that. And there is some percentage of those ticks in the south that are infected. It's probably much lower than in the north. My estimates are somewhere between 2 and 5%. And the adult ticks do bite people, and that's in the in the fall and in the winter and the spring in the south. So we do have some endemic Lyme disease from the black-legged tick in the south, and that's not really debatable. The question is, how much Lyme do we have here? But we have the conditions. We have animals that are infected. They are perfectly capable of infecting ticks. That's been demonstrated. And we have ticks that can transmit, and that's only dealing with the black-legged tick. So we'd have some Lyme just from that. Then there are all these questions, and it's very complicated. There's a lot we don't know still. For example, I still am not convinced that Lone Star ticks can never transmit Lyme. I mean, I think they may be able to, and we haven't done enough experiments with different strains of Lyme Borrelia to determine if they're completely incapable. But some people say Lone Star tick saliva kills Borrelia, but then the same people will accept that Borrelia Lone Stari, which is a relapsing fever Borrelia, is transmitted by Lone Star tick. So we know their saliva doesn't just kill all Borrelia. Maybe it only kills Lyme Borrelia, or maybe Lyme Borrelia don't exist in the same concentrations in Lone Star ticks, which makes it harder to detect it in them. And maybe they don't transmit it as efficiently because of that. So there's a lot of details, and the devil's always in the detail. When you make these broad statements that this never happens, this can't happen, that tick can't transmit this, and so on, it's a lot more complicated than that. And we haven't even come close to doing all the experiments that are necessary to prove conclusively one way or the other about some of those things. Dr. Hartman, I'll turn over to you and let you address.
Well, in my view, the biggest challenge for most people, actually, I would have to say there are two. The first is actually getting diagnosed so they can get treated. For many people, they have a wealth of symptoms and medical problems, but on average, by the time people end up seeing me, they've seen a dozen other doctors or more, doctors in all kinds of specialties. My mnemonic for Lyme is that you see pain, brain, and lame. People who have muscle pain, nerve pain, joint pain, brain problems, difficulty with cognitive function, fuzzy thinking, foggy thinking, attention deficit problems, difficulty multitasking, focus, concentrating, remembering, trying to lay down memory. And then some people will have other neurodegenerative disorders like Lou Gehrig's disease, Alzheimer's, Parkinson's. Some of those people actually have Lyme, multiple sclerosis. And then the pain, brain, and lame, people with chronic fatigue, people with fibromyalgia, rheumatoid arthritis, lupus, MS. And, sometimes people are physically lame. They will have enough neurologic deficits that they have difficulty with ambulation. So if somebody's got pain, brain, and lame, they deserve to be tested for Lyme. And that Lyme test deserves to be interpreted on the basis of not the CDC criteria, but, sometimes one band will be present. One antibody to Lyme is all the more that we'll see. I've seen people who came in with a history of a big red rash on an extremity, multiple doctors, negative Lyme tests, treated that person or those persons for a month, rechecked their Lyme test, and then they were CDC positive. So because of that immune suppression, and because Lyme has the ability to hide, people may not even have a positive test. I mean, I have a handful of people in my practice that it took several times testing before we found something that demonstrated that they actually have Lyme. And then there are other companies that you can do more specialized testing with. The one that most people know is iGeneX. My favorite one is Vibrant Wellness, where for $480, you can test for 15 different Borrelia strains and the major co-infections, Lyme, Bartonella, Babesia, Ehrlichia. And every person that I've tested for Lyme and co-infections with this Vibrant Wellness test, they all come back with various Lyme species and co-infections. So, I think the first biggest challenge is people don't get diagnosed. They have all those things going on and nobody can tell them what's wrong. And they've got problems in 10 different areas. And none of the specialists is looking at the whole elephant. They're looking at their little piece and, this is what it looks like. And well, we can't tell you what's wrong with you. You just are sick. And then the next challenge is it's not even easy to treat Lyme. In the past, we've always used rotating courses of different antibiotics. But in the last 10 years, there's been research published, a lot of it funded by Lyme advocacy groups, looking at what herbals are effective, establishing that there are different forms of Lyme, chronic persistent Lyme. Your average staph or strep is a fast growing organism that replicates on the order of hours. And antibiotics kill bacteria when they're in growth cycle. So if you have a strep throat and the strep replicates every few hours, you're going to kill off a major percentage of those bacteria. So 10 days of penicillin, you're going to get it all and they're going to be done. Well, Lyme can grow as slowly as replicating once a month. So even with the best regimen out there, it takes a year to get through a dozen growth cycles. So we have to look at Lyme differently. It has had millennia to develop mechanisms by which it can hide from the immune system. It can befuddle the immune system. It can interfere with the pituitary and thyroid and adrenals and female and male reproductive cycle function, produce toxins that end up giving people postural orthostatic tachycardia syndrome and mast cell activation. So it's important to understand that you can't fix Lyme with a 10-day course of antibiotics. And yet that's the standard treatment if somebody actually does get diagnosed with Lyme. They're given maybe a month if you get lucky, but that's not going to be good enough. You're going to knock it back, and then it's going to regrow, and then people are back in the saddle where they started. So in the last decade, with this new research that we've had, it's established that there is what's called a stationary phase of Lyme, Bartonella, Babesia. They're slow growing infections that are much more like leprosy and tuberculosis. And nobody would ever dream of treating leprosy or tuberculosis with a month of antibiotics, but that's what they're telling Lyme patients they should do. So people need longer courses of therapy with appropriate agents. And what we used to use when those various antibiotics like Zithromax, clarithromycin, minocycline, doxycycline, Cipro, tinidazole, metronidazole, when those are tested against these chronic, persistent, or stationary phase Lyme, Bartonella, and Babesia, they don't do so well. So the newest things that have come out that are highly effective for chronic, persistent Lyme and co-infections are things like dapsone, which is the leprosy drug, disulfiram, which is typically used for alcohol abstinence. It makes people sick if they drink with it. And something called methylene blue, which isn't a standard prescription in the United States, but I actually have my patients special order it or compound it. And those are things that are extremely effective for chronic persistent Lyme and Bartonella and Babesia. And then these advocacy groups have also funded what herbals are effective for chronic persistent Lyme, Babesia, and Bartonella. And interestingly, the herbal Cryptolepis was the most effective for every single one of those three. Japanese knotweed is highly effective for all three. My other favorite is Chinese skullcap, which does kill all three. And it's wonderful for helping with anxiety, insomnia, inflammation, joint pain, and histamine mast cell activation. So I love Chinese skullcap because it immediately helps people feel better, and it's treating the Lyme, Bartonella, and Babesia that's the underlying cause. And then I like to add in Japanese knotweed because it's a fabulous anti-inflammatory. It helps reduce inflammatory response in the brain. It helps with cognitive dysfunction. It kills the Lyme, Bartonella, and Babesia. And then I hammer in a big gun like Cryptolepis or dapsone or methylene blue, or I have some people that I will do a cocktail. I may do a blended regimen of herbals and prescriptions, or I may do just strictly herbals. But I love the idea of the herbals because if people can't get to a Lyme literate physician, they can do that herbal regimen. You know, all of the things in that are available to anyone without a prescription, without a doctor. So not only are there not very many doctors that are very adept with treating Lyme because it's extremely complex, but sometimes people are highly compromised in abilities on all levels, including their ability to generate an income and afford to be able to take the supplements and see an expensive doctor. So, you know, it's like you get a 1-2 punch. You know, people have to get diagnosed, and that's really difficult. And then they have to get decent treatment, and that's really difficult.
I first want to share that one of our audience members thanks both of you for your dedication and research and diagnosis and treatment. I think that everything that you both just shared about issues facing patients very much conveys your commitment to finding new information that will ideally better the experience of patients that are currently there. And in the future, hopefully we'll have less issues than is facing the current population. To come back to you, Dr. Hartman, and moving a little bit back into what you were speaking on in terms of co-infections, I wanted you to elaborate a little bit on the types of co-infections that you're seeing in your patients, as well as how you're working through that process of not only identifying Lyme disease, but also some of these other infections.
So ticks are like nature's dirty needle. When somebody gets Lyme, they don't just get Lyme. They get Lyme, Bartonella, Babesia, Ehrlichia, Mycoplasma, Q fever, rickettsial infections, Rocky Mountain spotted fever, Powassan virus, Heartland virus, microfilarial worms. I mean, ticks are like a microbial petri dish. The big three are Lyme, Bartonella, and Babesia. With Lyme, people tend to get severe fatigue because Lyme neurotoxins poison the mitochondria. They tend to have endocrine problems because Lyme poisons the pituitary gland. So that affects the adrenals, your ability to hang on to water. So people will have dehydration problems. It affects the typically female reproductive cycle function, and it affects the adrenals. And all of those are like rampant problems in Lyme disease. And then Lyme also has the ability through its neurotoxins to negatively impact the immune system so that the immune system doesn't go after it. Bartonella is a bacterial infection, and the commonly recognized form is cat scratch fever, where people will get a cat scratch and they'll get swollen lymph nodes above the area of the scratch. So it's common to see swollen lymph nodes, and Bartonella produces neurotoxins. So people will have anxiety, depression, irritability, nerve pain, neuropathic pain. You can also see joint pain with Bartonella, what we call Bartonella bulges, where somebody's got puffy fluid retention on the sides of the ankles, but they don't have edema. So, I mean, with Bartonella, a lot of what we see is neurologic, but you can sometimes see the swollen lymph nodes and something we call Bartonella striae, where people will have purple stretch marks, and they're not in the distribution that you normally see stretch marks, where, you know, there are stretchy areas in the skin. So that's something that's really pretty unique to Bartonella. Babesia is a parasitic infection that's sort of a cousin to malaria. And so with Babesia, people will see fevers, sweats, and chills. Now, not always do they have that. Sometimes they're just kind of hot and cold and hot and cold and hot and cold. You know, there's not like any comfortable temperature in between. They tend to swing between the hot and the cold, and they get worse at the full moon. In fact, all of these infections tend to hit growth cycle at the full moon. So people will have worse symptomatology at the full moon when their toxin load is higher. So with Babesia, fever, sweats, chills, and Babesia infects red blood cells just like malaria. And so that can make the red blood cells sludgy. And so if you have like sludgy red blood cells in the lungs, people can have chest pain, you know, pain that hurts when they take a deep breath or when they stretch. And it really is like little tiny microclots in the pulmonary circulation. When you see it in the cerebral circulation, people get headaches. Now with Bartonella, they tend to get like the nerves firing off, migraine types of headaches. With Babesia, they get pressure headaches because it's like everything sort of blocks off and they dam up their blood vessels and their cerebrospinal fluid, and they get pressure headaches. So diagnosing these infections is really difficult. One year I tested for Lyme along with Bartonella and Babesia in every person that came through, and I didn't get a single positive for Bartonella or Babesia. Part of the problem is there are multiple Bartonella and Babesia organisms that can cause disease, but we only have a test for a few. So unless we switch to more esoteric testing from specialty companies that's out of pocket, you know, it's going to be hard to identify those co-infections. You know, generally, I try to do it with a questionnaire. You know, I have people check off the symptoms that they have, and those symptom questions are grouped according to which co-infection that tends to correlate with. And so I just typically treat for what we see and then follow those symptoms as someone goes through their course of treatment. Typically, you know, if somebody's checking off 30 symptoms on a questionnaire and they lose 2, they might not notice that. But it's really nice to have that flow sheet because, you know, then we can come back and say, well, you know what, this one is better. This one is better. Or if somebody has new symptoms crop up and like, oh, you know, something else needs to be covered for it. Now, most of the regimens that I use are going to work well for all of the major big 3. And in fact, for others like chlamydia and mycoplasma and ehrlichia. So generally, the regimens that I use are going to cover for all of those other infections. So I pretty much assume that everybody has got all the big three and maybe a smattering of some of the others, and then cover for all of that. That way I don't have any nasty surprises.
Now, thank you, Dr. Hartman. Dr. Clark, I want to bring it back over to you. I know Dr. Hartman has mentioned several of the co-infections here, but I was curious if you could expand upon Borrelia americana and Borrelia andersoni and how they are involved in this whole process. Do they, create a different set of symptoms from Borrelia burgdorferi, and what should patients in the South be looking out for there?
Thank you, Noah. That's a good question, and I don't think I can answer it real specifically, but I will try to respond in a meaningful way. So one of the things that we know about Lyme Borrelia is that it's a species complex. So it consists of numerous species that are very closely related genetically, and there's well over 20 species in the complex now that have been described worldwide. There are several species within the United States. The most common one we hear about is burgdorferi. And then we've heard a lot about mayonii, which was identified in some patients up in Minnesota and Wisconsin. A little bit about one called Bissetti. It used to be called Bissettii. Now it's called Bissettii. That's been found in patients in California, and maybe, one patient that we described from Florida. And then there are others that like Andersonii and Americana and some others that have been found through molecular techniques in just a few patients. So we don't have enough data on enough patients to know or to see patterns in how they present. But I just kind of anecdotally, I expect that they may present in somewhat different ways than maybe the more common ways with burgdorferi. And by that, I mean maybe by more neurologic presentations with some, perhaps with Andersonii, and maybe more skin manifestations in some ways with Americana. And that's just from a few case reports. But I mean, I think the bottom line here, and Dr. Hartman touched on this, is that with a lot of these tick-borne infections, we have tests for only a few species or strains, and certainly that is true for Lyme borreliosis. We have tests that are mostly designed to identify burgdorferi and a fairly limited genetic group, or a group of strains for burgdorferi even. And we don't have data on whether the typical two-tier testing would even give a positive result person that was infected with Andersonii or Americana. We don't even know, not even through mouse experiments or something in the laboratory. No one has done that work yet. So we just don't really know. And that could, I'm not going to say that I have any level of certainty on this, but it could help explain why so many people with these Lyme-like symptoms do not test positive with the standard testing in the southern United States, because perhaps they are infected with genetically variable strains that are not what are more common in the northeastern US. And maybe we can get into this a little bit more somewhere along the way, but I just want to make it clear that for a lot of different tick-borne infections, but absolutely with Lyme, this is a dynamic situation in the country. Not only are you going to see differences from one region to another because of different animal hosts and possibly different Borrelia strains and so on, but it's a dynamic situation. It's changing all the time. Ticks are moving in different directions. We have Lone Star ticks that are moving further north, not for the first time, regardless of what you read about Lone Star ticks now moving to Long Island because of climate change. Actually, Lone Star ticks used to be present in the northeastern United States all the way up to southern Maine, and then they disappeared for a while because of various human manipulations of the environment and almost eradicating white-tailed deer, and now they're moving back up there. And likewise, black-legged ticks from the northeastern United States are migrating down along the Appalachian corridor, and they're becoming established in the mountains of North Carolina. And I fear that they may continue to move further south along that corridor. And before you know it, they're going to be, if they're not already, in eastern Tennessee, and they're going to be maybe in northwestern South Carolina, and then they're in northern Georgia, and they may go all the way down to Birmingham, Alabama, because that's how far the Appalachian mountain range actually goes. And so then it's going to be constantly changing. Not only do we have whatever situation that currently exists in the southeast, but it's changing all the time. And then we're going to have northern populations of ticks moving into the south. Whether they're ever going to meet and eventually mix completely, I don't know. And what's going to happen when that happens, I don't know. But it's fascinating, but it's also extremely frustrating because we have so little knowledge about all of this right now.
So when I have patients who get some of this more specialized testing, like for instance, the vibrant wellness testing will actually give me the species of Borrelia. And many of my patients have multiple Borrelia species. Like I had one who had Borrelia turricae, which is a tick-borne relapsing fever species, and Borrelia burgdorferi, Borrelia, some European strains, Guerinii, Afzelii, Spielmanii, and Bavariensis. So I've had people that have had, four species of Lyme organisms all present, and then multiple co-infections as well. So when there are that many variables, it's just hard to know what's producing what symptoms. And I'm not familiar with any data identifying, different Lyme species and differentiation of symptoms that you would see. So to the best of my knowledge, the same treatment that works for Borrelia burgdorferi works for all of these other Lyme species. But I agree with Carrie that the Lyme Western blots that are done are cued to the northern species of Borrelia burgdorferi. And so that if people have a different organism, their test may not be positive. So it's no wonder that the testing is such a crapshoot, because you just can't, you can't pin it down. People have symptoms and the tests don't pan out.
Can I follow up on that just for a second? Because
Yeah.
one of the things about the testing, because it lacks specificity and it lacks sensitivity, which sensitivity is probably the biggest issue when it comes to Lyme. Often people will report that they have one or two bands on a Western blot. Like maybe they'll have a screen test that's equivocal, and so then they'll have a Western blot and they'll have maybe only one or two bands. And inevitably, if they have only one band, it's usually the 41 band, which the 41 is just a number that coincides with the molecular size of a particular protein called the flagellin gene. So that's what that represents. And all Borrelia have a 41 kilodalton protein flagellin gene. So, a major question is, are these people who show these 41 bands or only the 41 band, are they infected with just some type of Borrelia, a cross-reactive positive for some other closely related organism? It's possible, that's been demonstrated possible, but we just don't know. And, just as an example, Dr. Hartman mentioned a couple of Borrelia species, for example, the relapsing fever species, the Borrelia turricae. But to get back to Borrelia lone star eye for a second, because Noah, I think you brought that up a minute ago. When people talk about lone star ticks and possible Lyme, again, we have to keep in mind that there are many different organisms out there in these ticks. And there are still major questions about Borrelia lone star eye, because if you just put Lyme aside for a moment, Borrelia lone star eye falls in the relapsing fever category, and it's one of only a few relapsing fever Borrelia that have been found in what is called a hard tick. You may have heard of the Borrelia miyamotoi. That's one that's been found in the black-legged ticks as well. But in the Lone Star tick, they found this one that they named Borrelia lone stari. And it's very common throughout the distribution of the Lone Star tick. On average, maybe 2% of all Lone Star ticks seem to have this relapsing fever Borrelia. Yet there's virtually no evidence of it being in humans. So the question is it just not pathogenic? Because let me tell you, people are being bitten by so many Lone Star ticks, they are being exposed to this Borrelia. But is it infecting people? Is it making them sick? Could it be possible that it has an affinity for the brain or something like that? And so it leaves the bloodstream and doesn't act like other relapsing fever Borrelia. And maybe that's why it's so hard to detect. We don't know, but I will throw this out there. And by the way, analogy is a really weak form of evidence. I realize that. But there aren't many Borrelia that we have identified anywhere in the world in any type of human biting arthropod that is not pathogenic to people when people get exposed to it. There are Borrelia that exist in nature in cycles with, say, a tick species that never bites people. Okay, so that people probably won't ever get exposed to that one. But when you think about Borrelia miyamotoi is a relapsing fever one transmitted by the black-legged tick. Lyme Borrelia, there's relapsing fever that are transmitted by body lice in Africa and places like that. So I'm not aware of any Borrelia out there that comes into contact with people that hasn't been determined to be pathogenic to people so far. And so we don't have a lot of evidence. I'm just saying, let's keep an open mind about this. We don't have specific antibody tests for Borrelia lone stari. And we've done very little work to determine whether or not that could be a potential human pathogen. And instead of just concluding that it's a of minor concern and it's self-limited and that kind of thing, I think a lot more work really needs to be done on that particular species because it could possibly explain a lot of the Lyme-like illness in the southern United States. And at least it's a logical thing to pursue because we know there's tons of Lone Star ticks. They bite people more than any other tick down here. We know that a certain percentage of them have this Borrelia species, and it's very similar genetically to the Miyamotoi species. So anyway, I just thought I'd throw that out.
Well, the other thing that I want to say is our testing isn't good enough that we can say for sure somebody does or doesn't have Lyme. And so my rule of thumb is if it looks like a duck and it walks like a duck and it quacks like a duck, treat it like a duck. Don't just assume they couldn't have it because there's very little harm in treating. And if somebody improves with treatment, there's a good chance that we're hitting the nail on the head there.
So I'm looking at our Facebook Live real quick, and I see a very interesting question here that I think helps to move this conversation along. Is that somebody mentioned that we continue to speak about ticks, but clearly there's other vectors out there as well. They had heard that they could potentially get infection from a housefly or a mosquito. I don't know the exact answer to that right off the top of my head, but I figured that either of you might be able to provide at least some direction as to is that an accurate claim that they've heard, or is that something that still needs additional research? What do you both know about that?
I'm not familiar with any. Oh, I'm not familiar with any data in the literature that says that mosquitoes or houseflies can transmit Lyme.
And what I would say is that there may be a few published papers that show evidence of some Borrelia in mosquitoes. I think there may be some evidence from Europe or China or both where they did report that. I haven't seen any definitive evidence on their ability to transmit. And just taking the most conservative approach there in terms of evidence, it's inconclusive. But I will say this, I've talked to so many patients over the years who have experienced so many different illnesses or symptoms after having contact with different types of biting arthropods, and maybe not a housefly, but biting flies, maybe you know, these no-see-ums, these gnats that people often, they'll call them sand flies or sand fleas sometimes, and they're actually, most of the time, these are biting midges. More often referred to as what we call no-see-ums, where they're so tiny you can't hardly see them, but if you look close enough, you can actually see these things. They're very common along coastal areas, along beaches and things. And what I would just offer is that, you know, I don't discount people's reports of what their experiences are. You know, sometimes they maybe try to connect dots that can't be meaningfully connected, or maybe we just don't understand what's going on. But you know, there are so many different things that can cause the same symptoms. And the symptoms for different tick-borne and other arthropod-borne infections are so similar, as you know, things like fever and body aches and fatigue and so on. And so I would not say that they couldn't have some arthropod transmitted infection. It might be Lyme, it might be something else, because I think there are many things out there in nature that can cause these similar symptoms. And you know, and you treat with some broad spectrum antibiotic, perhaps like doxycycline, and the patients get better, and often you don't know what they actually had, but if they get better, they get better. You know, that's the ultimate goal from a clinician standpoint. It's an art form. It's not a pure science. Treating patients is not a pure science. It's an art form, and you learn by experience, as Dr. Hartman knows. So there are so many things out there in ticks, and the more you look, the more you find. And I continue to find more organisms in ticks. And Dr. Hartman mentioned Babesia and things like that. There are patients that have symptoms that are very consistent with babesiosis. And there are Babesia in different tick species in the South that have never been fully characterized. And we don't know if they're potential pathogens to human, but I know that they're out there because I've been finding them in different ticks, like Lone Star ticks. There's some Babesia species that we found in a percentage of Lone Star ticks. It's associated with white-tailed deer as well, and it may or may not be a human pathogen. We don't know, and we don't have tests for that. Rickettsia, and I don't mean to go on too long, but Rickettsia is something that I've been looking at and testing a lot of different tick species. And, you know, almost every tick species has some type of Rickettsia in it. They may not all be pathogenic to people, but we know, for example, that Lone Star ticks can be infected with a particular type of Rickettsia. Actually, they can be infected with a couple of different types, but one is called Amblyommatus after the Lone Star tick. That may be a potential pathogen, at least a mild one. The Gulf Coast tick transmits Rickettsia parkeri. That's been proven conclusively. American dog ticks don't just transmit the one that causes Rocky Mountain spotted fever. They are infected with multiple Rickettsia species across the Southeast, and I think several of them may be human pathogens, at least mild pathogens. And then black-legged ticks may even be able to transmit some rickettsia that cause human disease. And so that's just another whole group. And so when these patients present with these symptoms and they've never been treated before, and you treat them with these broad-spectrum antibiotics and they get better, who knows if what they actually had. But yeah, lots of different pathogens and lots of different arthropods, and people don't learn new things.
The other thing that I would say, I mean, I made a rather bald statement that I'm not aware of anything in the literature. Now that's not, I mean, the absence of proof is not proof of absence. It's conceivable that somewhere down the line, there may be established proof of other biting insects transmitting. We don't have any data on that now. The other thing that I want to say is ticks the size of a poppy seed can crawl up, attach, bite, infect, drop off. And if it's really tiny and it's in an unobtrusive area, that person has no idea that they were bitten by a tick. So, there are times when it may look like it was caused by something else, but they actually had an inapparent tick bite. So, I don't base a decision to treat on whether or not I have a history of a tick bite. If somebody has the symptoms and I have some corroborative data, I treat. And I don't care where it came from. They need treatment.
Awesome. Thank you both for your in-depth answers. I hope that for those who had questions on our Facebook Live, that addressed some of those concerns. I do, for the sake of time, want to kind of move to some concluding statements here. So I will pose a very general question that ideally will, showcase how we as a community can look to move forward in addressing these issues that are facing patients. I would say, what do you see as the biggest opportunity for, Lyme nonprofits or researchers to pursue in order to, work to dispel some of these issues? Be it research, be it, whatever you may see. And I'll start with you, Dr. Clark.
Thank you, Noah, and I appreciate you answering that, asking that question. So, we think we know a lot about Lyme disease and some of the other tick-borne infections, particularly in some areas of the country where they've been studied pretty intensively. For example, the northeastern U.S. is where we've done the most research, and they're still learning new things, dispelling all sorts of myths up there. For example, the white-footed mouse being the most important reservoir. Now it looks like maybe shrews or something like that is more important in infecting ticks, but yet, we thought we knew what the most important animals were. In the Southeast, we've barely scratched the surface on these things, and things could be so different down here than in other parts of the country. And of course, I live in the Southeast, I do research down here, but I can't help but feel a little bit left out a lot of the time, that, people just, we're forgotten down here. It's almost as if, nobody really cares. The numbers aren't that high. They think we don't really have it here. And I understand we care most about what's in our own backyard. Everybody does. That's fully understood. But I think that there's a lot more that can be learned by particularly focusing on some of the areas that have not been studied as intensively. And the more you look, the more you're going to find. That's everything practically that I've ever looked for as far as a disease organism in ticks or animals, I have found in the Southeast. And the ecology may be different and the epidemiology is different and so on. But if you don't look, you're not going to find those things. And I'll give you a quick example of something else that I'm studying right now. It may turn out to be non-important as far as human disease, and that's an organism called spiroplasma. Spiroplasma have emerged in some papers in the recent years as possibly causing human disease. It may be another tick-borne disease that's transmitted by different tick species. Spiroplasma have never been found in any ticks anywhere in the eastern United States. Well, I've been testing ticks just in the last couple of months, and we're starting to find some spiroplasma there. I don't know if they're human pathogens or not. And there's virtually no funding to do this, but I'm experimenting as much as I can. So one aspect of the research is just knowing what's actually in the ticks in this part of the country, and that can possibly help inform what's going on with some of the patients. If we know what's in the ticks, then we can possibly translate that to better testing for human patients and maybe figuring out what's causing some of those types of diseases. So some more comprehensive work, but across the Southeast, because it's a huge region and there's tons of biologic diversity throughout the Southeast, and things can be, pretty different in different places.
So from my perspective, because I'm a clinician, I want to continue to see studies that tell us about agents that are going to help us eradicate Lyme and co-infections. So, for me, that's the number one area that I would like to see more research on. Now, I think my second, if I got to have a second choice, my second favorite would be research that allows us to identify ways to interrupt the life cycle of ticks so that people don't get bitten. You know, one of the things that's happened in America is, you know, people moving out from the cities and out into more rural areas that you now have small pockets of areas with trees where deer and white-footed mice and shrews, things like that, are going to prosper. And so the Lyme prospers along with those organisms, of those kinds of animals. And so as people kind of invade into nature, but break it up, now there aren't a wide variety of different kinds of animals that can survive in little pockets of treed areas. And so it reduces the biological diversity. It tends to be restricted to animals that perpetuate the growth cycle of Lyme. And then now, you know, we have people like in pockets around Atlanta or around Middleburg or around, you know, where I see huge concentrations of people with Lyme. So given the biodiversity of organisms and animals in an area, it can predispose to have very high concentrations of Lyme in animals that can live in small pockets of wooded areas. So if there are ways that we can interrupt that, then we're going to have fewer Lyme organisms and fewer ticks that can bite people. So, you know, that would be a wonderful thing. But first, I want to know more research that's going to identify things so that I can get people better.
I appreciate both of you for your time today. I think, as to reiterate what one of our attendees said, both of your work has helped at least make some progress for patients, and I look forward to seeing the future of what that work may look like. For all of our attendees, I know there were some questions that we were unable to get to today, either from the Q&A on the Zoom or those watching through Facebook Live. So if you still have questions, I would encourage you to send an email to info@projectlyme.org, and we will do our best to get back to you as soon as we can with an answer that ideally will help you move through with your own case, or to be able to better convey information to those that you may know who are interested in learning more about Lyme and tick-borne diseases. Again, thank you both, Dr. Clark and Dr. Hartman, for taking the time today. And I wish you all the best of the evening.
Thank you. Thank you for having us.
Thanks, Noah, and thanks to Project Lyme and for some of the funding that I've gotten recently. That's amazing. I really appreciate it.
Oh, great.
Of course, we look forward to working with you further. I know Bay Area is on this as well, so shout out to them for their work on the research side of things. And everybody have a great evening. Thank you so much.
All right. Thank you.
Thanks.